So I'll start with a job description. I'm a university professor in a med school, so the University of Florida College of Medicine. Being a university professor, typically the job is described as being divided up into three areas: research, teaching, and service. The way my job is structured now, I think I've spent about half my time on research and probably 25% of my time on teaching and 25% on service. Teaching in a medical school, being a professor in a medical school is a little different than being a professor in sort of a liberal arts college or something like that. In a medical school, the students that we deal with are professional students, including medical students and dental students and pharmacy students, depending on the medical school, and we also deal with graduate students. What's important is that we don't deal with undergraduates. That's a good news, bad news situation. The good news is that it means that there's probably half the teaching load relative to being in a liberal arts college. The bad news is that you don't interact with undergraduates. But the impact of this is that what it means is that you have typically much more time for research. You're also in a clinical environment, and what that means is that in general there can be on average a much more robust research environment in a medical school because the professors have more time for it and because it's interfaced more intimately with a clinical environment. So it's a terrific place to do research if you like doing research. If you would prefer to be teaching undergraduates, then you're better off in an environment that's not a medical school, but it makes it more difficult to do the research. At any rate, so I get to spend about half my time doing research, and I'll come back to exactly what that is. In general, what it means is that you have to define a research problem, write a grant, get the money, set up a laboratory, populate it with students and postdocs and etc., and if you're lucky you get time to actually play in the lab. And you have to publish papers describing your research down the road. For teaching, I teach medical students, I teach graduate students, I teach, have taught pharmacy students occasionally. I teach entirely in my area of specialty, and it really amounts, my teaching load is relatively heavy, and it amounts to about 30 lectures a year, which is, as I said, about half what you'd find in a liberal arts college. As far as training for this job, I think the single most important thing is to do some research when you're an undergraduate to find out whether or not you like this. That's what happened to me. I worked as in a laboratory as a sort of an independent study when I was an undergraduate, so I was 19 or 20 years old. I worked in laboratories for both my junior and my senior year. I think doing that much is appropriate to find out if you like it. And I just fell in love with the environment. I liked the laboratory, I liked the whole laboratory environment, I liked dealing with the science, and my role models were the professors, and I decided then that this is what I wanted to do. I wanted to be a professor and have my own laboratory in a university, and I was able to follow up on that goal. Not everybody is that focused, but I would say that unless you work in a laboratory and discover that you like it, you could go to graduate school and discover that you don't like it, and then you got a problem. I think that's the main motivation. After graduate school, you can figure on doing a postdoc, which is a pure research thing in somebody's laboratory for 3 to 6 years, and then you're eligible for a job in a variety of different markets, including an academic market like this. The job market is competitive. If your goal is to get a job and have plenty of money and not have to worry about that stuff, and that's your major goal, I don't think this is where you ought to be. I think you do this because you want to do this, and you can anticipate perhaps some hassles in getting a job down the road. I've had 10 PhD students and 3 postdocs. All of them are in science in some capacity or another. A lot of them have said that they want to be in academic science, and none of them are at this point. One of them is a research assistant is an assistant professor at the Scripps Institute, but he's going to have to leave pretty soon because his grant money has run out. He's looking for a job and he's having a difficult time. I don't want to make it sound like it's impossible, because I think if you're good, you can do anything. But I don't want you to go into this with rose-colored glasses thinking that there's going to be plenty of jobs out there. The market's competitive. If you're good, you'll get a good job. As far as salaries go, they actually pay you to go to graduate school because you're a research assistant working in somebody's laboratory, and you can anticipate between $20,000 and $25,000. They pay you more as a postdoc, more as a university professor. I also list as a possible option biotech, because if you're interested in research, you don't necessarily have to be a university professor. All my students, most of my students, are in biotech companies of one sort or another. And you can see from the slide what the salary ranges are. In general, you can actually support a family on a single salary, though some supplementation from another salary or Santa Claus would help out. But you can do okay. These salaries are listed, the top end about $150,000 in each. That's assuming that in each biotech or the university, that's assuming that you're not going to go on and be a VP or a dean or a chairman, in which case you're going to make more money. I really like the casual lifestyle, and that's one of the things that attracted me to this initially. In an academic environment, you have significant independence as to what you want to do in terms of research and how you want to use your time. You have opportunities for international travel. I got friends all over the world that I've never worked with or never lived with. They're just professional associates that, matter of fact, 5 or 6 of them are coming in February for the second annual pox virus golf invitational tournament. Okay, and we're all going to play golf for 3 days. These are just people I've known for 20 years because I go to meetings with them, and they're wonderful people. You will hear that the discipline requires long, weird hours. Okay, people talk about being dedicated to spending nights and weekends and all kinds of stuff. Frankly, I have issues with this. I've been able to run my life basically with a 40 to 50 hour week, and I pride myself in being very efficient and very organized, and also leading a balanced life. So I am an example of the fact that that can be done. A lot of people put a lot of emphasis on sort of a macho type of dedication to the discipline. That's fine if they want to do that. Don't let it scare you away, because it doesn't have to be that way. What's hot in the disciplines now? I've listed a bunch of things on the slide: genomics, bioinformatics, biodefense, emerging diseases. If you're into virology, is a big deal. Virology comes into gene therapy because they use virus vectors to deliver genes. Neurobiology, stem cell research, that's a little different than what I do, but it is in the biomedical research arena. Those are the major areas where there's some excitement going on at this point. My research, I work on the molecular genetics of vaccinia virus. Vaccinia is the virus that was used as a smallpox vaccine. This has had some press lately because of the potential of smallpox to be used as a bioterrorist weapon. But in fact, my interests are entirely basic, and I've been working on this virus for 27 years. Okay, and nobody, except thankfully the NIH, at least a little bit, has cared a whole lot. But I really enjoy it. It's a wonderful model system for looking at the regulation of gene expression, and I'm interested in the viruses themselves. For anybody who doesn't know, smallpox, as shown in this slide, was a devastating disease that presented itself as a blistering skin disease. Everybody got it in the mid-1700s in Europe and actually all over the world. As a childhood disease, same way as you get chickenpox nowadays, and 25% of the time you died. So it was not fun to live with. And if you survived, you were seriously scarred. In the late 1700s, an English physician named Edward Jenner figured out how to vaccinate people against it. And let's back up. After talking about smallpox, I want to show the next slide, which shows the structure of the virus. It's a fairly unusual and complex structure, and the next slide shows an infected cell showing how the virus assembles in the cytoplasm. So it's a very visual sort of life cycle, though we do a lot of molecular biology, et cetera, to study the life cycle. In the late 1700s, an English physician named Edward Jenner figured out that you could introduce from cattle a disease called cowpox into the skin of people who had never been infected with smallpox using this bifurcated needle shown in the next slide. Actually, the needle was invented later during the smallpox eradication campaign. But basically, you put a little bit of live cowpox virus into broken skin on an otherwise unaffected individual, and as shown in the smallpox vaccination slide, you get a single blistery lesion on your arm and no other disease beyond that, except in rare cases where there are complications, and that confers immunity to smallpox. That technique was established ultimately worldwide, and the disease of smallpox was eradicated. My research doesn't focus on any specific health aspects, but more the fundamentals of how the virus grows, how it replicates. In general, what I've done is to put together a large collection of mutants of the virus so that I, in effect, have an on-off switch on each gene in the virus. I can then infect a cell, turn off the gene, the virus is no longer working properly, and I do a lot of molecular biology to look and see exactly what's wrong with the infection, and by understanding what's wrong, make deductions about what that gene ordinarily does. So it's basic stuff having to do with the replication of the virus. Let's do the good news and bad news about my job. Okay, let's start with the bad news, what I don't like. Actually, there's not much I don't like. I feel privileged to be able to do this. There are some things I'm not really crazy about. I don't like writing exams. I don't like sort of forced creativity. In order to write a good exam, you got to be very creative, and that's always kind of a chore that I procrastinate about. I'm not crazy about grading exams, but there are some upsides to that because you learn what the students know and what they don't know, and that's an instructive experience. I definitely don't like stupid committees, and there are plenty of them around. Over time, you learn which ones are stupid and which ones aren't, and if you don't like stupid committees, you can say no and not serve on them. I've had my dealings with administration. I respect people who do it, and it's not for me. I don't enjoy it, and so I avoid almost any type of administration at this point. What I especially like is the one-on-one interaction with students in the laboratory. Watching them learn, interacting with them, teaching them stuff one-on-one, learning about their lives is just absolutely wonderful. I enjoy lab work, though frankly I don't get to do much of it anymore. I'm pretty much tied to my office with other activities, the teaching, grant writing, reviewing papers, directing people in the lab sort of on a remote basis. I enjoy understanding how living things work and being a part either directly or indirectly in surprising new scientific discoveries. As I've said, I enjoy the casual and independent lifestyle, and I do enjoy classroom teaching as well. In retrospect, I'd do it all over again. I don't think I'd change anything. I've really enjoyed the career. I've experienced a significant amount of stress in the process of getting here, but I don't think that was really necessary. So I would learn how to manage the stress at a younger age, and I think I could get to exactly the same place and enjoy it a lot more. That was my fault, nobody else's fault. Once again, I would recommend it to anybody. I know I've said a few things that sound perhaps a little discouraging, but I don't want to discourage people from this because I think it's a satisfying and rewarding discipline, and we need people to do this kind of stuff. But it's also, as I've indicated, can be challenging. So you've got to be up to the challenge, and the way to test yourself in that is to find a laboratory to work in as an undergraduate and work in there for a significant period of time. There are laboratories at the University of Florida all over the medical school. Not many people know that, but you can find them. There's a great interdisciplinary program through the biochemistry department that advertises a significant number of laboratories. But you can also just find them on your own, and there are also laboratories in IFAS and the College of Liberal Arts and Sciences, etc. Just go knocking on doors and find a place to work, and